Hello everyone and welcome to this video content of the plant biology practical work which I teach at University Mustafa Ben Boulaid, Batna 2. In this practical work, we plan to use long-term field experience as a teaching tool to enable life science students to achieve a number of important pedagogical objectives. One general objective is to encourage students to acquire a greater plant culture through specific practical experience, as well as to realize and appreciate the role of scientific research in understanding the nature world. For the introduction, this course or practical work introduces the formal concept of the science of plant biology and the impact of plant on man and the earth. Topics include plant structure, composition, growth, diversity, life cycle, and natural history. The world's major plant environment and the economic influence of plants on our species. Students gain a better appreciation of the importance of plants in our life and are introduced to the fundamental concepts used in the study of plants. In the laboratory section, students learn the basics of science. The practical work of plant biology is divided on three practical work. The first one is plant cell, the second one angiosperms flower, and the third one is the primary anatomical structure of the stems. For the first practical work, intitulated Plant Cell, the objective is microscopic examination of various cellular components by the optical microscope, allowing for the observation of the cell wall, nucleus, plasma membrane, cytoplasm, and vacuole. For plant material, the red onion cells are suitable for this experiment because their colored shape make observation easier. The processes of plasmolysis and deplasmolysis demonstrate the movement of the water in and out of plant cell due to difference in water potential. About laboratory material and reagent. In this practical work, we used the optical microscope, distillate water, saline solution, forceps, glass slide and cover slip, glycerin, razor blade, filter paper, watch glass, neutral red, and the Ranger's solution. The procedure 1 is the collection. We cut a fragment of epidermis from the concave side of the fleshy skull of an onion bulb. About experimentation, the first observation of the plant cell in the fresh state. There is the observation in the optical microscope. The second one is the observation after staining with safranin or natural red. The third one is the observation of the red onion cells after saline solution treatment. After that, we pass So, the second practical work, intituled Angiosperms flower, the objectives are investigation of morphology of angiosperms, focusing on the roots, stems, leaves, and flower, with an emphasis on both monocotyledonous and dicotyledonous plants, gaining knowledge about the various flower compounds and the overall organization of a complete flower. Then exploring floral diagram and understanding their representation. After that, examining floral formula to better comprehend the floral structure. The plant material is the mustard plant, which is any one of several plant species in the genera Brassica in the family Brassicaceae. Mustard seed is used as a spice. For the laboratory material, we use a razor blade, binocular loop, forceps, and a black plate used for arranging floral compounds for observation under the binocular magnifying glasses. For the experimentation, so we observe the compound of the angiosperms flower, which is the mustard plant. Here there is the diagram flower of the angiosperms flower, which is the mustard flower or the mustard plant. For the third practical work, which is the primary anatomical structure of the stems, the objectives of this practical session are to compare a monocotyledon stems with the dicotyledonous stems. Specifically, we aim to observe and analyze the tissue types present in each, including the epidermis for surface covering, parenchyma for general support, carenchyma, and sclerenchyma for structural reinforcement, and phloem and xylem for fluid transport. Additionally, we will examine the presence and characteristics of secretory tissue, such as epidermal hair. The laboratory material and reagent, we use plant material, distillate water, acetic acid, carminogreen, forceps, razor blade, optical microscope, glass slide and cover slip, watch glass, and the bleach. For the proceeding, A. Sampling. Using a razor blade, thin transverse cuts are made on the stems, which is monocotyledonous and dicotyledonous. B. Staining and mounting. The method involves a series of steps for preparing and observing stem sample under a microscope. Initially, thin transverse cuts are made on both monocotyledonous and dicotyledonous stems using a razor blade. These cuts are then transferred to a sequence of watch glass containing specific solution. First, the samples are treated with bleach for 15 to 20 minutes to destroy cellular content. Next, they are rinsed with distillate water for 3 minutes to remove any residual bleach. Subsequently, The samples are treated with acetic acid for 5 minutes to eliminate rimming, bleach, and leaving matter, while also promoting staining through neutralization. After this, the tissues are stained using a staining solution for 3 to 10 minutes to enhance visibility under the microscope. Finally, excess staining is removed by rinsing the sample with distilled water. The prepared samples are then mounted on slide with a drop of distilled water and covered with a cover slip to avoid air bubbles formation. Observations are made under the microscope at both low 10 and high 14 magnification. Here there is the observation of monocotyledonous tissue, which is in the left one, and the observation of dicotyledonous stems, which is at the right. And thanks for your attention.